Think forward. Think Research Channel.
It's a real delight to be here tonight. First of all, I would like to thank the Academy for doing these talks to the public about what's happening at our university, introducing the public to our faculty and the wonderful science that is occurring there. I think this is critical for the university to be able to reach out to all of us that live here and for all of us to appreciate what a gem we have in the University of Wisconsin-Madison. As Mike said, I'm Managing Director of the Wisconsin Alumni Research Foundation, otherwise known as WARF. WARF is the patent management organization for the University of Wisconsin-Madison. And for those of you who know me, one of my loves is patents. I'm passionate about them. And the one reason that I'm passionate about patents is that it allows me to rub shoulders with great people. And one of those great people happens to be Hector DeLuca. In fact, Hector, in my mind, and I'm sure I'm not alone in this, is one of the great scientists to have trained, worked at, and come out of the University of Wisconsin-Madison. I'm going to briefly review some of Hector's accomplishments, and then I'm going to tell you about the Hector I know. First of all, Hector grew up in the Arkansas Valley of southern Colorado, a son of a farming family. And he is a farmer by nature. When I first knew Hector, he was growing potatoes up in northern Wisconsin. He received his bachelor's degree with honors at the University of Colorado. Then he became a research assistant at the Department of Biochemistry here at the University of Wisconsin, working in the laboratory of Professor Harry Steenbach. Harry was the founder of WARF, and Hector happens to be the last graduate student of Harry Steenbach. Hector received his master's degree in 1953 and his PhD in 1955 from the Department of Biochemistry. He directed Professor Steenbach's laboratory for 2 years, and then he was offered an instructorship in biochemistry from Conrad Elvium, then chair of biochemistry and dean of the graduate school. Hector then spent 1 year of study at Strange Ways Research Laboratory in Cambridge, England, learning to do tissue cultures. When he returned, he rapidly progressed to associate professor in '63 and full professor in '65, and was awarded the new Steenbach Research Professorship of Biochemistry, an endowed chair that he holds today. In 1970, he was named chairman of biochemistry, and except for a 5-year vacation, he calls it, between 1985 and 1991, he continued as chairman until July 1 of last year. During Hector's tenure of biochemistry, it became really one of the great biochemistry departments of the world, and it continues that. I think it consistently ranks in the top 5. And again, like being proud of the University of Wisconsin-Madison, what Hector has done with biochemistry is phenomenal. In a state of 5 million people, to have the type of science that goes on there. I'm told that we have more National Academy members in the biochemistry department alone than many colleges have. Hector advanced the study of vitamin D at all levels. He first discovered that vitamin D must be converted into the body into a hormonal form. And it's really his work that was seminal in allowing people to understand that vitamin D is a hormone. It's his work that unlocked and explained what the structure of the active form of vitamin D was. And this work was done back in the early 70s. And it was the first time that WARF really stepped outside the United States and entered into some major license agreements with Japanese companies that brought in a lot of good royalty money to the university. His organic chemistry training led him to the synthesis of a large number of analogs of vitamin D, and today he's working on the 4th and 5th and 6th generation of the active forms of vitamin D. His most recent contributions have been specific analogs that simulate the formation of new bone, stimulate the formation of new bone, now a candidate for a pharmaceutical market. Hector's been responsible for the formation of at least 3 companies in town. Bone Care International, which was recently purchased by Genzyme. Tetrionics, which is out at the Research Park and was purchased by Sigma Aldrich. And his own company now, Deltanoid, which we've got great hopes for. And you know, he's not our youngest entrepreneur, but he's probably our most successful entrepreneur, one of our most successful entrepreneurs. He's received many awards for his contributions. Among them are membership in the National Academy of Science, the American Academy of Arts and Sciences, the Wisconsin Academy of Science, Arts, and Letters, the Rousseau Prize of France, the 3M Life Sciences Prize, award of the Federation of American Societies for Experimental Biology, the Bristol-Myers Squibb Mead Johnson Award for Distinguished Achievement in Nutrition, and on and on and on and on and on. He's received an honorary doctorate of medicine from the Karolinska Institute of Stockholm. Swedes? More recently, he's been named the Ernst & Young Entrepreneur of the Year. I like this Young Entrepreneur of the Year award. And he and his group have been credited with at least 8 pharmaceutical products still being marketed worldwide. And now, very quickly, I just want to tell you about the Hector I know. Hector's Wharf's most prolific inventor. He has in his portfolio well over 200 U.S. Patents. He has cornered the market in using vitamin D for end-stage renal disease. And I'd like to tell you that Hector's inventions have brought back more royalty money to the University of Wisconsin than all of our inventors combined over the 80 years that Wharf's been in existence. But more important than that, his discoveries have treated millions of people and made the lives of patients better. Let's see. I've had the privilege to know Hector for 21 years, and I consider him a friend. I know him first as a very loyal individual. He's loyal certainly to his family, and he's got his family here, Margaret Claggett Dame, who is not only his life partner, but his business partner, and thank God she's his business partner. And his kids, Martin and Adriana. He's been loyal to the UW-Madison all these years, and he's incredibly loyal to Wharf. He's passionate. Hector has that zest for life, learning, and discovery. It's a great model for all of us. Excuse me. He has a mind that's forever young and engaged. He meets every Tuesday at Wharf with our staff to talk about new discoveries. And, you know, it is amazing to watch the interaction of Hector with the young staff and how he instructs them. He's at base a teacher. He's a tough negotiator, and I've had to be on the opposite side of the table with Hector. There are times I wish that I could employ him at Wharf to do the licensing for us. And finally, he's an Italian. You know, Italian includes all the above. It includes passion. He can be excitable, and I've been the subject of that. But he's always charming, and so I give you Hector DeLuca.
I just don't know what to say after that introduction. I thank you very much, Carl. I think some of it was true, anyway. I'm really grateful for that introduction. I want to thank the Wisconsin of Sciences, Arts, and Letters for this opportunity to talk with you about something that's been very dear to my heart for for all my career. And I want to also show how the relationship has evolved between the university, vitamin D, and WARF. And so the title that you saw originally is true, but I want to also now give you a subscript title. I also want to thank all of you to take the time out of your busy lives and to spend this valuable time with me this evening. So thank you for coming, every one of you. And this now tells you what I want to discuss. And I want to show you how the intimate relationship between WARF, the University of Wisconsin, vitamin D, and how it's an open-ended story that will continue, we hope, long into the future. Now, this is an old story. It began quite some time ago. And I want to start with the discovery of the vitamins, which really played a key role in vitamin D discoveries and WARF at the University of Wisconsin. You have to imagine that in the middle of the 1800s, organic chemistry had flowered in Germany, and the German organic chemists dominated science for a long time. And they spilled over into the field of nutrition, where they held very firmly that an adequate diet could be adequately described by measuring carbohydrate, protein, fat, and salts. And that's all that was required to support life. And yet there were occasional investigators, such as Lunan and Magendie, that would feed purified components of the diet according to the German chemist's idea, and the animals would fail to survive. But that did not cut a hole in that armor, and this idea persisted for some 50 years. Now, where the armor really began to crack was here at the University of Wisconsin in Madison. In 1883, the university hired Stephen Babcock. Let's see if I've got a pointer here that works. Who was actually the second biochemist, but really the true father of biochemistry at the University of Wisconsin. And his passion was nutrition. He was very much against the German concept, but he could do nothing to overturn it, because he had no animals with which to carry out experiments. The animal husbandry department here had the dairy herd, and it was for show. These were beautiful animals, and the chairman of animal husbandry did not want experiments run on those animals. So Stephen Babcock did the next best thing. He perfected the famous Babcock butterfat test, which really started the Wisconsin dairy industry on a firm basis, and he's really quite famous for that early measurement of milk quality. But finally, a new chairman of animal husbandry came in by the name of Humphrey, and he began to appreciate the value of experimental science. And he listened to Babcock and turned over the dairy herd to Babcock for an experimentation. By that time, Babcock was near retirement, so he hired Professor E.B. Hart, who was chairman for some 40 years of biochemistry. But he started, and he in turn hired a very young assistant professor by the name of Elmer McCullum, and they carried out this single grain experiment, largely the idea of Babcock. And Babcock reasoned that he was going to divide the dairy herd into groups of four, and he would feed the first group only wheat, the second group only corn, and the third group only oats, and the fourth group a mixture. And he made sure that those diets all had the same amount of protein, carbohydrate, fat, and salts. And they observed these animals. The animals on wheat did very poorly. They couldn't produce milk. They aborted their young, and some of them died. Those on corn did extremely well, were very productive, and oats were in between, and the mixture did very well. They correctly concluded that this illustrates that simple measurement of these macro components cannot describe a diet, because there were substances required for life and reproduction that were yet to be described. And so the idea of the German chemists began to be cracked. At the same time, Casimir Funk carried out an experiment like Magendie, where he again lost animals when they were fed a purified diet, and he thought there were some very vital substances, which he called amines, that were required in the diet, but really couldn't prove it. And so it was E.B. Hart who turned Elmer McCullum loose on, let's find out what these components are that are required in the diet. And this is truly another brilliant investigator that I think formed the Department of Biochemistry in those early days. He decided, and rightfully so, that he couldn't do his experiments on cows because he would have to mix enormous amounts of diet. He would have to shovel enormous amounts of poop. And so he reasoned that he had to work with a smaller organism, and so he chose the pet rat. Well, you could imagine what that did in the College of Agriculture, housing the enemies of a farm. So Babcock said, listen, just hide him in the basement of biochemistry. The dean will never know, he never comes down here. So that's exactly what he did, and so he first trapped wild rats, and they were so mean he couldn't work with them. So he personally went to Chicago and bought 2 breeding pairs of white or albino rats and started what is now known as the Sprague-Dawley strain of experimental rats. And so he bred them in the basement of Biochem and set up experiments that he could carry out. So very brilliantly purified the protein, the carbohydrate, and he left milk whey as a source of salts, and then he varied the fat, and he found that lard did not support health of these animals. They developed eye lesions, wouldn't grow, and he then found that butterfat and really cod liver oil had something in it that really made these animals grow. And so he discovered what he called factor A, fat-soluble factor A. He didn't even call it a vitamin in those days. Well, that caught the attention of the scientists throughout the world, and he then did another clever experiment, which he published a couple of years later, in which he replaced the milk whey with weighed pure salts, which he took off the shelf, and he found that they developed, and they fed cod liver oil to give them the factor A. He found that they developed a different disease, a neurological disease, and some of them died, and he called this water-soluble factor B. Finally, the German chemist idea of diet was destroyed. McCullum, in talking with Harry Steenbach, who was an undergraduate that worked in the department on the single grain experiment and became a faculty member, decided to rename it, taking Funk's vital amine idea, renaming it vitamin A and vitamin B. And similar work was now being done by Mendel and Osborne in Connecticut. And so the whole idea that there are vitamins that prevent disease, this was the hot subject, just like stem cells are today here. This was the hot subject, and so everyone began thinking about the diseases that might be nutritional diseases. And that's where the vitamin D story had its beginning. Sir Edward Mellanby in Britain was really taken by the fact that 50% of the children in Scotland, and even greater numbers or percentages in England, developed a serious bone disease called rickets, and 10% of the children died of a convulsive tetany. And it was in epidemic proportions. In fact, the Germans called it the Englische Krankheiten, or the English disease, because England was a low sunlight country. It had undergone the Industrial Revolution. Smoke filled the air. Children were moved into cities and were deprived of sunlight, and unknown to them, vitamin D was not produced in their skin. So Mellanby decided that this must be another nutritional disease. So he inadvertently fed dogs a diet of the Scottish people, oatmeal, and kept them indoors. And sure enough, they developed the same disease as children, serious rickets. Oddly enough, he found that if he gave them cod liver oil, he'd been reading McCullum's papers, he could cure this disease. And it was logical to conclude that vitamin A had another activity to cure rickets. Well, that was, in the meantime, I want to tell you a story about McCullum in a couple of minutes. But in 1919, at the same time that cod liver oil was curing the rickets in dogs, Holczynski in Vienna, a physician, happened to take rachitic children outdoors in sunlight and cured their rickets. He also then irradiated them with ultraviolet light, and it also cured their rickets. So this really strange dichotomy evolved in which light equaled cod liver oil. Now, how could that make any sense? And that's where Steenbach was the brilliant discoverer that put this whole thing together. But I've got to tell you a little side story before we get to that, and that's about Elmer McCullum. Elmer McCullum left Wisconsin in 1918 to go to Johns Hopkins University, because that Hopkins was like a command. If you got an offer from there, it was a command. It wasn't an offer. So he accepted the job there, and just want to show you that these people, though brilliant scientists, were human. Before he left, he turned all of the white rats loose in biochem, empty the cages, and the reason is he didn't want any competition in discovery of the vitamins, because E.B. Hart was going to turn over the rat colony to Steenbach, and he just couldn't feature that, so he turned the rat. So there's a big discussion in the science, if you go back and read those journals, between E.B. Hart and McCullum at Hopkins, arguing about ethics. So these guys are human. No, they want their credit as well as anybody else. So, but McCullum is truly a brilliant investigator. He saw the cod liver oil and he thought, that doesn't sound right. So he bubbled oxygen through cod liver oil. That destroys the vitamin A. And he found that cod liver oil in which he destroyed the vitamin A still cured rickets. He said this is another vitamin, which he called it vitamin D. We're missing C. That was now being discovered at another institution. This is where vitamin D first had its birth as a vitamin, which is actually a misnomer, because vitamin D is not found in our diet as a rule. We make it normally in skin by ultraviolet irradiation. We're fortunate that we can absorb it and we can take it in diet, but it was discovered and called a vitamin when in fact it's a prohormone that's made in your skin. Okay, so what about getting back to the cod liver oil and sunlight idea? Steenbach had, when McCullum was working with white rats, he was assigned the goat colony. So he worked with goats. In 1916, he noticed that if you took the goats outside in the sunlight in the summer and he measured calcium balance, he noted that they kept their calcium in their bones. But when he took them indoors in the wintertime, they began to lose calcium. So somehow sunlight had something to do with calcium. So when the McCullum and Sir Edward Mellanby's observations began to click in place, Steenbach realized that ultraviolet light is doing something, and he showed very clearly that you could irradiate food, skin, a number of biologicals, and produce an activity that would cure rickets. And so he discovered the irradiation process for producing vitamin D, which made possible the isolation and identification of the chemical structure in 1932. But more important than that is this, that he eliminated rickets as a major medical problem throughout the world because that irradiation process allowed individuals to briefly irradiate milk and other suitable foods, impart vitamin D, and rickets disappeared as a major medical problem. An enormous contribution to the health of the United States and the world. But Steenbach realized this was an important discovery for a lot of reasons. One, it could be abused, and sure enough, Steenbach told me that one of the things to his shock was the appearance of a label on cigarette packages that says, fortified with vitamin D by the Steenbach process. He said there was a carload of beer in Milwaukee ready to be shipped, which got stopped by an injunction which they were able to get, where beer was fortified with vitamin D by the irradiation process. So he realized that you could abuse an invention, and therefore there had to be some way to protect it, and the patent system has one such advantage. I mean, you can at least keep people from using it in the wrong way if you choose to do so. The other thing is, of course, it's worth money. And this was an unpopular thing for anyone to do in those days. For a professor who's supposed to be pure to patent and get this dirty commercial money. But he did it with his own money. He went to Chicago and found a patent attorney like Carl Gulbrandsen. He wasn't Norwegian, though. And paid for the patent, and then he came back and presented it to the dean of the College of Ag and Life Sciences, Dean Russell, I believe, who took it to the administration, and Wisconsin took the enlightened position that an invention belongs to the professor. Okay? But they did it for the wrong reason. I love Wisconsin because it's the only school that has what should I call it? Anyway, this view that the invention belongs to the inventor. And they did this because, as Dean Russell put it, we don't want dirty commercial money coming into the university. Has that ever changed? Thank God. So it was actually with considerable bravery that Steenbach continued to persist. He got $1 million from Quaker Oats for a license fee, which he put in the bank. He didn't touch. and he kept trying to see if he could get the proceeds of his invention to support further research at the university. When that didn't happen, he came up with the idea of a foundation, and together with Dean Slichter, Dean of the Graduate School, they came up with the idea, which is now the Wisconsin Alumni Research Foundation, in which they got, I can't remember, I think it's 5 or more alumni who each contributed $100 to start the foundation. And I guess the $100 of each of them is still at Wharf. And then they gave their time in helping direct this foundation. And this foundation, I got to say, has made Wisconsin different from all other land-grant colleges. It raised it another order of magnitude, and it remains as an extremely important arm and element of this great university. Without it, we could not be what we are. And as I want to point out, Wharf has provided hundreds of millions of dollars over the years to the University of Wisconsin-Madison to support research in all fields. The other interesting thing is, as Carl alluded to in his introduction, Wharf is the forerunner of intellectual property management at universities by 50 years. It wasn't until the late 60s that most other universities even got interested in this. And Wharf is the template which is used to pattern intellectual property management at universities, I think, throughout the world. So we're really grateful for this generous and visionary concept of Steenbach and to Wharf. Well, this is where the story now shifts to me and shifts to what has happened since I came to the University of Wisconsin. When I went away to the Strangeways, I realized they had Strangeways, and I decided I'd come back and work at Wisconsin. So, when I was away for that year, I thought about vitamin D, and we don't know how it builds better bones, but it would be a wonderful thing to try to find out, and maybe in the course of learning that, we'll figure out how to treat diseases. So one of the first things was to chemically synthesize radio-labeled vitamin D, and guess what? It disappeared before anything happened, and we learned that vitamin D, as we know it, as we take it in our vitamin pills or we make it in our skin, is inactive. It doesn't work. Instead, the body has to process it, and I know most of you don't care about these little squiggly structures. I have some chemists over here that do, but the liver has to process this and put a flag on it on the tail, and now it becomes the blood form. Then it becomes recognized by a system in the kidney that hangs onto it and puts another group to form the final active hormone. And this step is very tightly controlled. As you need calcium, it's turned on. If you don't need calcium, it's turned off. And so that constitutes a beautiful endocrine system. And it is this form of vitamin D that makes the body absorb calcium or redistribute it and phosphate, and makes it possible to mineralize and grow bones. And so the primary effort of this hormone is to raise blood calcium and to make calcify bone. And we've got to remember that this probably was one of the early developments of this hormone, but because it's present, it took on a number of other functions. And these are functions that are more minor and take a second seat to forming bone, but to us now they're becoming very important because they are a clue as to how we can treat other diseases with this interesting molecule. And so that's a theme I want to set before you. Okay, so the first thing we did was when we first isolated and identified and synthesized the blood form. At first we thought it was the active form, and we got a lot of activity. Roussel of France right away licensed it. It was also the patent that broke the federal hold on patents from federal funding. And that's another story which I don't have time to go into, but that led to the institutional patent agreements that allow organizations like WARF, once again, to patent inventions made with federal funds, and led to the Bayh-Dole Act, which made it mandatory that federal grants allow nonprofit organizations to hold patents discovered with federal money. So this was used right away to treat the bone disease that results from kidney failure. And we'll talk a little bit more about that in a minute. And that's one of the major applications to some of the active forms of vitamin D. This happens to be a child that was born at UW hospitals that had Russell Chesney's patient. He's 3 years old, and he was on dialysis from day one, and he had severe rickets, severe bone pain. The poor kid would cry just to move. And then he was treated with our first synthetic vitamin D hormone. And this is the boy 14 months later, and he survived for kidney transplant, and then everything was okay. So right away, it's very rewarding. The discovery itself is very rewarding, but add on to that the benefit to people like this, and you start thinking that's a pretty good reward. Okay, so as we continued, this lists some of the things that went on. We isolated, identified the final vitamin D hormone. That was quite a job. We made it chemically, got licensed, and these are still pharmaceuticals that are sold today. And we made analogs, and this is the one that Carl talked about that is used in Japan even today to treat osteoporosis, in Southern Europe and almost every part of the world except the United States and Northern Europe. And we'll talk about why that's true. We also discovered then the vitamin D endocrine system and how intimately it was controlled by calcium and how it in turn caused the mineralization of the skeleton. I want to finish these off. Another important event for Wisconsin is the synthesis of 1-alpha-hydroxyvitamin D2, which is Hectorol, which is licensed to Bone Care. We started a new company, and you heard about that. The other interesting thing is that in competition with another major group, we cloned the vitamin D receptor. Now, this is the protein that recognizes the vitamin D hormone and makes it work in the cells. And this becomes important as far as realizing that there are other functions of vitamin D. So just to give you a brief idea of how the vitamin D hormone works, I want to tell you that it binds to this receptor, this protein that recognizes it, and it causes certain genes to express their protein, and these are the proteins that then make calcium transport or do some of the great things that vitamin D does. So as we begin to learn that there might be other functions of vitamin D because the receptor is found in places we never thought that it would be found, Dr. Tatsuo Suda in Japan made a very important discovery. He is a former student of ours, and he found that if you add the vitamin D hormone to cancer cells in culture, that they stop growing and they differentiate into functional cells. And so this is a function quite apart from building bone and mineralizing the skeleton. It focused now that vitamin D might do some other important things. This excited a lot of people because maybe vitamin D compounds could be used for differentiative therapy of cancer, but so far that hasn't really come to fore. I'm going to go backwards. I think that I went too fast here. Hold on just a second. The other important finding is that vitamin D could control proliferation. And so it was a natural thing to look around at possible places where proliferation could be corrected by vitamin D. And the keratinocyte is a place where we find the vitamin D receptor. The keratinocyte is a skin cell, and it, under conditions of psoriasis, will multiply and rapidly grow and proliferate. So scientists, not our group, found that, in fact, if you treat this disease topically with the vitamin D hormone, you can markedly improve this disease. And in fact, Leo Pharmaceuticals has on the market Dovanex, which is a successful topical treatment for psoriasis. So here it is, vitamin D, not just for bones anymore. Okay, so how about multiple sclerosis? A location of the vitamin D receptor is in the immune cells. Well, what's it doing there? It has nothing to do with calcium absorption. It has nothing to do with with bone. And so we begin thinking about diseases related to the immune system. And multiple sclerosis is one of those diseases. It is an inflammatory disease in which antibodies are made against the central nervous system proteins that then destroy the central nervous system and give the progressive paralysis of multiple sclerosis. So with Margarita Contorna in our group, we decided to look at the immune system because we had other evidence that vitamin D might play a role in the immune system. And so she came up with the idea that we ought to look at the model of multiple sclerosis, and it's called experimental autoimmune encephalomyelitis. And this disease can be produced in susceptible mice by injecting into these mice protein that's isolated from the central nervous system, myelin basic protein. And if you do that to these mice and add to it a little pertussis toxin, you start getting the disease. The first thing that happens is you get a drooping tail. Then you start getting paralysis of the hind limb. It moves into the forelimb, and eventually you have to sacrifice the animal because they're unable to move, and it goes in a sort of relapsing situation. If you give the vitamin D hormone, now this is above the amount made in their bodies. If you give the vitamin D hormone, you can prevent this from occurring. Okay, why don't we treat multiple sclerosis with this active form of vitamin D? The reason is the vitamin D hormone, its preferred mode of action is to raise blood calcium. Hypercalcemia or high blood calcium is extremely dangerous. And in fact, a sidebar is don't ever take too much vitamin D because you'll calcify your kidneys. Okay? It's a potentially dangerous material when taken in large amounts. So you cannot treat multiple sclerosis with the vitamin D hormone because of the danger of destroying kidneys and other soft tissues and due to vitamin D intoxication. Experimental animals, you get away with it, and it works. Okay, so interesting, but no cigar. Let's look at the osteoporosis situation that we talked about earlier. That, isn't it strange, in low calcium countries like Japan, Italy, southern France, you can use the active hormone of vitamin D to treat osteoporosis with considerable success. The fracture rate falls to half of what it is otherwise. It does work. Why don't we use it in this country? We eat lots of calcium. We are at 800 to 1,000 milligrams per day. With all that calcium, the danger to hypercalcemia or high blood calcium is very real. And no drug house wants to produce a compound that's dangerously close to being toxic. So it can't be used in a high calcium country. So, and you'll remember that the disease osteoporosis is a disease of bone mass. What happens in the case of postmenopausal women where it's really exaggerated, but as a function of age? The honeycombing or intertwining of the internal parts of bone are the most labile, and they erode away. They disappear as you use that calcium up to support other aspects of life. And if you look at the honeycombing in a normal bone versus an osteoporotic bone, you can see that the girder support is gone, and that results in crush fractures, which you get loss of height. It's painful, but even more serious are the hip fractures, long bone fractures, which generally end up in elderly in serious mortality. So what is our approach to this? Here we have a bunch of possible diseases that we should be able to treat. Vitamin D is doing something. So our answer is, let's take the molecule, and we've learned over the years how we can modify it and produce some beneficial effects. We've learned how to reduce the ability to raise serum calcium. One of our drugs, Zemplar, is such a drug, which we developed in 1993 and is on the market to treat renal patients because it has a bigger window between hypercalcemia and treating the bone disease of kidney failure. So by applying these modifications that we've learned, we came up unexpectedly, Rafael Sachinsky in our group learned that if we remove a grouping from here and put it here, and we change the configuration of this carbon from the natural to the unnatural form, all of a sudden, this compound favors this very small activity of regular vitamin D hormone to build bone. And so now we have a bone-specific form of the hormonal form of vitamin D. There are other possibilities. We've learned that we can change this molecule and change what it does. It will activate certain genes and not others. And so that is the dream and the hope of the kind of work we're doing today. So let's look at what 2MD does. If you take human osteoblasts in culture and You incubate them for a period of 2 weeks. And won't get into the goodies that you add. This is, you see osteoblasts, but no really formed bone. If you incubate them with the vitamin D hormone with really very heavy doses of the hormone, way more than you'd ever see in the plasma, you begin to see a little bit of activity. If you give even small amounts of this new analog, enormous formation of bone nodules occurs. So that was one of the cracks in the dike. And when we gave this compound to rats that have had their ovaries removed, they're retired breeders, they already have lost a lot of bone, you could build that bone right back. So this drug, 2MD, is currently in phase 2 trials in osteoporosis. Every now and then we have a little setback, but we're very confident at Deltanoid that this compound will be in the market, will be treating osteoporosis, and we'll be doing it in a physiologic way because what it is an anabolic agent that builds back bone. It does not block the breakdown of bone, which is what most of the drugs that are available now are doing. This is to show you the ovariectomized experiment that I was telling you about. This is the way a bone looks like in these rats that have been retired breeders. They're usually of about a year and a half old. And you take out their ovaries, so they've lost every semblance of estrogen. They lose all of the honeycombing inside the bone. The shaft becomes thin, and the green stain shows you bone. And you notice that in the magnification of that, they're very small specules of bone. Here's the bone after treatment with a dose of 2MD that does not cause hypercalcemia. It does increase blood calcium somewhat. But you'll notice now, this is what's happened. We've built back a lot of that honeycombing. The shaft is very thick with bone. And so it looks very, indeed, promising that this is the only drug that we know of that will both build back this kind of bone and bone shaft at the same time. So we have great hopes for it, but again, there's no cigar yet. I'd like to talk about another autoimmune disease that we've played with in our laboratory, and this is type 1 diabetes. Type 1 diabetes is another autoimmune disease in which antibodies attack and destroy the islet cells that produce insulin. I'm sure you know how serious this disease is and how debilitating it is and how troublesome it is even when managed. So there are models of it, and we worked with the NOD mouse model, non-obese diabetic mouse. And what happens is that they spontaneously develop the disease. And I want to tell you that if you make these NOD mice vitamin D deficient, they develop it more quickly and more severely than if you give them ordinary nutritional vitamin D. But even more important is if you give them the vitamin D hormone, you can block the disease completely with the expense of hypercalcemia. Again, that problem of serum calcium. Now I want to sit back and tell you a little bit about the importance of ordinary vitamin D nutrition and supplementation. And some people would like to say exposure to sunlight. A whole bunch of autoimmune diseases, there's colon and prostate cancer, all show the same distribution of disease relative to hemisphere. Near the equator, these diseases are all at low incidence. As you progress north or south of the equator, the incidence of the disease becomes greater and greater. It's been suggested, but not proved, that it's vitamin D that's the protective agent. And in fact, experimental models such as this support that idea. Furthermore, there's some belief that the amount of vitamin D that we use to protect our skeletons from rickets, and in our cases, osteomalacia, which is the adult counterpart of rickets, is insufficient, and that we should be on higher intakes or higher production rates of vitamin D. In Wisconsin, we make no vitamin D in the winter. The sunlight comes in at an inappropriate angle, and it does not convert 7-dehydrocholesterol to vitamin D. So we're essentially deficient of skin production of vitamin D from sunlight, from the end of November until sometime late in April. Some scientists are recommending that you expose yourself to sunlight, and some scientists are saying that's bad because of the danger to skin cancer, especially melanoma. And we have a very good alternative. Oral vitamin D, we absorb it easily. Why risk getting melanoma? So we believe the intake of vitamin D should be increased up to about 2,000 units a day, and as high as 5,000 units a day is probably safe. Anything above that you shouldn't take unless a doctor directs you to it, because you could be one of the sensitive ones and go into hypercalcemia fairly easily, and hypercalcemia, vitamin D intoxication, is no picnic. You want to stay away from that. So vitamin D is potentially dangerous, although it is beneficial at these low doses. So I want to put in a plug for adequate vitamin D intake to help you against some of these autoimmune diseases and certain types of cancer. Okay, so what do we do about the hypercalcemia? And I want to conclude now by showing you another situation where vitamin D therapy is clearly needed, and that is kidney patients. Obviously, kidney patients are lacking the vitamin D hormone because they don't have any kidney mass anymore. But what happens is that in the absence of vitamin D, of course you don't absorb calcium. You can't mobilize calcium from bone at physiologic doses. And we learned much later in our discovery that the vitamin D hormone also plays an important role in shutting down the parathyroid, because the parathyroids, when there's not enough calcium, will continue to proliferate and run out of hand, and what keeps them in check is the vitamin D hormone. It suppresses this parathyroid activity, and that's a normal occurrence. But in the absence of kidneys, you lose this ability to absorb calcium, which is a big insult to produce parathyroid. And furthermore, you've lost the feedback regulation by the vitamin D hormone on the parathyroid gland. And this parathyroid literally chews up the bone. And these patients on dialysis suffer severe bone disease. And for a long time, and continues even today, vitamin D hormone and its analogs, one of the chief applications is prevention of this disease of dialysis patients. But again, all along we're playing with the problem of hypercalcemia versus therapy. And so we've been developing analogs where we breed away the ability to raise calcium, and hoping that we can get an analog that we can treat these diseases without hypercalcemia, and I can tell you that we believe there's a lot of hope. And for example, we have a compound that we've completely removed the side chain on, and this compound, when given at significant doses, will very markedly shut down the parathyroid, but it does not change serum calcium. And so a whole series of these compounds have now been made, and we're starting to apply them to a number of diseases. So that brings me to the last slide, and that is, what are we doing now? And this is through Deltanoid Pharmaceuticals and the university where the discovery is being done. The patents are filed through WARF, and the development is done by Deltanoid Pharmaceuticals. We're really putting as our number one priority to get this 2-MD to the osteoporosis patients. We know it works in animals. It's a very good drug. It's anabolic. It's going to grow both cortical and trabecular bone. We think it's going to be an important adjunct against disease in our aging population. We have made analogs that are even better than 2-MD with a wider window of therapy for this, that have the property of building bone, and those are under development. We want to treat psoriasis in a systemic way. Right now, the only thing out there is topical application. There is no oral drug for psoriasis. And we think by taking one of these non-calcemic analogs, we'll be able to give it by mouth and essentially eliminate the plaque psoriasis. We want to develop a better drug for dialysis patients, so it's a lot safer. These patients have so much going wrong with them, the last thing you need to do is give them a drug that endangers them to hypercalcemia. And now, the autoimmune diseases. And here are the ones that we're targeting: multiple sclerosis, inflammatory bowel disease, Crohn's disease you've heard of, type 1 diabetes, and if we get good non-calcemic analogs, there are possible anti-cancer drugs in it. So, to conclude, vitamin D therapy is not just for bones anymore. We're looking at a lot of other indications. We think we can get there through the vehicle of modifying this beautiful molecule to make it do what we want it to do and not do some of the bad things. Thank you very much for listening.
Thank you so much, Professor DeLuca. Professor DeLuca will now take some questions. We do ask that you raise your hand, and Bart Sanford and I will try to get a microphone to you as quickly as possible. So, do we have any questions on this part of the room? And when you ask your question, please speak directly into the microphone because of the taping going on.
I have a cardiologist who really pushes vitamin D. Could you tell me what effect it might have for the cardiovascular system?
The question is, does vitamin D have any action on the cardiovascular system? Carl Gulbranson has a patent on heart muscle and vitamin D. It turns out there's no receptor for the vitamin D in heart muscle. However, in the blood vessels, the intima of the blood vessels, there is vitamin D receptor, and there's very likely to be a role for vitamin D in blood vessels, and not necessarily on the heart muscle itself. Having said that, there are also secondary reasons why it might be beneficial, because you're supplying in a constant supply some of the minerals that are used by heart muscle to function properly. Other than that, those are the only reasons I can think of at the present time.
Any questions on the other side of the room?
Dr. DeLuca, is there any, what causes hypercalcemia?
Okay.
Did I miss that in your lecture? If I did, I'm sorry.
Yeah.
Can you just repeat simply what causes it?
Yes, the question is, what causes hypercalcemia? Vitamin D, the active form of vitamin D, turns on the small intestine to absorb calcium against a concentration grade. In other words, it will concentrate it from the intestine into the bloodstream. If you don't have enough calcium that way, it will work together with parathyroid to mobilize it from bone. And a small amount is good. It raises blood calcium to normal. That's required for mineralizing the skeleton. However, if you overdo it, you pump too much calcium from the gut into the blood, you mobilize too much calcium from the bone, you raise calcium above normal. Now, blood is normally supersaturated with calcium and phosphate. That's the state of events that's needed to support neuromuscular function and needed to mineralize skeleton. But if you go above that, it makes blood a metastable solution, and you start wanting to crystallize that calcium and phosphorus out where places where you don't want it. Kidney is a favorite site because there you're concentrating urine, you're making it even more concentrated, and you start depositing mineral in kidneys. Then you begin to block the function of the kidney tubule, and you start getting kidney nephrosis. You start dropping calcium in the heart muscle, blood vessels. You start dropping calcium even in the subcutaneous part of your body, which isn't as dangerous as it is in the blood vessels. So when you start calcifying too much of your tissue, you start destroying function. It's going to be lethal.
So what do you do to bring it down?
Well, there are a lot of things. If you have hypercalcemia, besides looking for the reason for it, first you got to suspect vitamin D intoxication, primary hyperparathyroidism. But if someone comes in and presents with hypercalcemia, doctors will usually try washout procedures, will give you a diuretic to make you, bring it down into the normal range. And you can even be treated with glucocorticoids or cortisone type drugs that will block the action of vitamin D and block calcium transfer. And that brings it back into the non-dangerous range. But it is, our body normally has a protection against hypercalcemia. There's another hormone that's secreted from the thyroid. It's called calcitonin. And when blood goes too high, it triggers the secretion of that hormone. And one of the things that this hormone does is goes to the bone and stops the osteoblast from pumping calcium back into the bloodstream from bone, and that brings it into the normal range. So there are a number of drugs that can be used to help you against hypercalcemia if you detect it soon enough. Of course, you want to get the primary reason. If you can't come in with hypercalcemia right away, someone's going to order a measurement of 25 hydroxy vitamin D in your blood. They're going to measure, look at a measurement of parathyroid hormone, and right away that's going to tell you two very possible reasons why you've got hypercalcemia. If it's vitamin D intoxication, you're going to go on diuretics and a glucocorticoid. If it's parathyroid, you're going to look and see whether you have a primary adenoma and are you a target for surgery. That's that's the other option.
Any questions on this side of the room?
What role do you think that vitamin D plays in depression or prevention of depression?
Ooh, everyone got that question? You're asking, does vitamin D play a role in depression or prevention of depression, right? I don't know that there is such a function of vitamin D. Some of the retinoic acid that is used to treat acne has been blamed for some, adverse events like that, but I'm unaware of vitamin D being used to counteract depression. So I can't give you a mechanism because the receptor is found only in very unusual places in the central nervous system. So the cerebrum is not one of them. So I can't give you a mechanism. It's possible that if you're deficient in vitamin D and tend to hypocalcemia, that the low blood calcium contributes to a lack of feeling good. Whether it would cause depression or not, I don't know.
Any questions on Barb's side of the room?
Did I understand you correctly that for persons who have type 1 diabetes or other autoimmune diseases that many scientists would suggest that taking between 2,000 and 5,000 milligrams of vitamin D would be a safe idea and a good idea?
The question is, people with type 1 diabetes, is that what you're asking? The question is, if you have type 1 diabetes, would anyone suggest taking higher doses of vitamin D? The answer to that is no. If you have type 1 diabetes, your islet cells are already destroyed, okay? There's nothing you can do about that anymore. So that's not going to help you against that disease. If you know you're going to go into type 1 diabetes, you have genetics that say your chances are pretty good. If you take the higher doses of vitamin D, it might lessen your chances of developing the disease. That's about all we can say right now.
Any questions over here? I think we can take two more.
I was wondering what the physiological differences are between Hectorol and 2MD.
Okay, the question is, what's the physiologic difference between Hectorol and 2MD? Hectorol is what's called a prodrug. It has to be acted on by the liver to produce the final hormonal form, which is 1,25-dihydroxy-D2. That compound does not have anabolic action on bone to any degree. It's sort of like the 125 slide I showed up here, where there's a little bit of effect on bone, but not a lot. We don't know the actual molecular mechanism why they're different. Okay, but in fact, if you give Hectorol and try to build back bone mass of the ovariectomized animals, you just can't do it. Whereas with 2MD, even at low dose, you build back that bone mass. So there's something going on that this analog activates in the bone cells that is not reached by either 125D3 or 125D2, and hence Hectorol. So I think Hectorol, we actually made that in 1974 for the first time, and then we got a good synthesis for it in the 80s, which made it possible for bone care to develop it. And it originally was going to be used for osteoporosis. That was the aim. But I think the road to osteoporosis treatment was so long and expensive that the people running the company decided that they would treat bone disease of kidney failure. And it does work. It does work there, but I don't think it'll ever be used as an anabolic agent for treatment of bone.
Another question on the other side of the room, and then I think we'll wrap up.
There's a gentleman there that's had his hand up quite a few times. I don't know.
Vitamin D appears to be a treatment for a lot of different diseases. Is there any known common bond between these diseases?
No, as I was trying to tell you, vitamin D is not a treatment for all of these diseases, but the survey data indicate that in places where there's higher vitamin D production or intake, the incidence of those degenerative diseases is lower. Now, what we're trying to do is develop a modification of vitamin D that can be targeted for that particular disease. We're trying to do that, but haven't achieved it yet. Am I getting through to you? I'm not sure.
I guess I was wondering if there was a connection with these diseases. There's something common that...
Many of the autoimmune diseases are common. The question is there any connection between all these diseases that vitamin D could be used to treat, if I understand your question right? The answer is yes. In the case of autoimmune, there is a thread. You're trying to suppress the inflammatory reaction that's occurring. In all cases of those, it's the inflammatory disease that destroys your tissue. And what you're trying to do is suppress the inflammatory reaction. And vitamin D appears to do this at the expense of hypercalcemia. So again, what we're trying to do is breed out the hypercalcemia but retain the ability to treat the immune cells. Okay? In the case of cancer, the common thread is anti-proliferation, and it holds that in common with psoriasis. That's the only thread. And so there may be some common genes that determine this proliferation, and that's probably what we're targeting, but I couldn't tell you what those genes are.
I think one last question, and then that's it. Anything on this side of the room? Oh, you're so far. Maybe Barb can handle you.
Bottom line, what do you recommend for the general public for a recommendation for vitamin D intake?
Your question is, what is my recommendation for vitamin D intake for the general public? I'd say 2,000 units a day. That's safe. I think no one will go into hypercalcemia with that dose. And it's about, about 4 times higher than the current recommended daily intake. It's possible that you could go higher, probably 5,000. I've seen one report of 2,000 units causing hypercalcemia in a very sensitive patient. Whether that's really true and supportable, I don't know. But when you get near 5,000 units a day, I think you got to start worrying. And I think you've got to do that under observation by a physician who will check your blood calcium periodically.
Professor DeLuca, thank you so much.